Hi, Michelle Glass here. Welcome to our final installment of our Chapter 22, Lecture 22, Part 1 series. And our topic here are what are described as your non-specific defenses. Now, your non-specific defenses mean they are generalized. They are not identifying what the pathogen is. These are just mechanisms that our body has to prevent pathogen invasion or identify that the cells are foreign, and as a result of that, attack. So we're not identifying what the pathogen is, the name of the pathogen, anything along those lines. And these are defenses then that are innate. These are things that you're automatically born with. So you don't have to be exposed to pathogen in order to produce these particular defenses. We have a whole list of non-specific defenses. So real quick, I think, yes, we have 7. So we're going to be looking at each of these in turn. So we have physical barriers, phagocytes, immune surveillance, which we've sort of mentioned a little bit about already, interferons, complement system, inflammation, and fever. So these are the 7 that we're going to talk about in this video. So to begin, let's think about our physical barriers. Now this is kind of like Anatomy 101. We talked about the job of the cutaneous membrane, creating a physical barrier that prevented pathogens from gaining access to your body. Ta-da! That's a physical barrier. So that's what we're talking about. We see that very often your physical barriers are epithelial tissues, so either lining the outside or the inside of the body. And we see too that a lot of times they involve some kind of secretion. So if with your cutaneous membrane, you have some antibacterial properties of your sweat secretions and your sebum. Also, it's making a watery surface, so the pathogens have a hard time adhering. When we're talking about your digestive tract, one of the benefits of your stomach secreting hydrochloric acid is that it will denature and break down any pathogens that you might have consumed. We talked about the pseudostratified ciliated columnar epithelium of the respiratory tract producing a mucus that will trap pathogens, and then the cilia will beat it down into the stomach so the hydrochloric acid can get rid of those pathogens. So that's 2 different physical barrier systems working together. So we see this quite a lot. So that's pretty straightforward. Okay, next we have phagocytes, which we've been talking about phagocytosis again from the very beginning of the course. Your phagocytes are considered your first line of cellular defense because it is their job to move through the body and identify that the cells are foreign. They don't know their name, who or what they are. They just know they don't belong, and then phagocytize it. So phagocytes include white blood cells that we already talked about in chapter 19. So your neutrophils and your eosinophils are going to be part of this group. We also have what we can describe as, we can describe them as macrophages, the neutrophils and the eosinophils. And then we also have the macrophage, which we've talked about, pretty much from the beginning of the course. Those are, of course, remember, derived from monocytes, which are another one of our white blood cells. Now, these are all different from lymphocytes because this is non-specific. So your macrophages and macrophages, they don't know who or what it is. They just know it does not belong. And as a result of that, they're going to phagocytize it. Okay, so the macrophage can actually take 3 different courses of action to get rid of our pathogens. One is phagocytize it and then use lytic enzymes from the lysosome to break it down. Another is to kind of neutralize it by binding to it and removing it from the interstitial fluid and then waiting for some other phagocyte to come along and take care of it. And then the third way is the release of toxins. So there are 3 listed here: tumor necrosis factor, nitric oxide, which we've talked about a little bit previously. The abbreviation there, remember, is NO, or hydrogen peroxide, H2O2, all of which can be toxic to invading pathogens. Okay, the next type, so we've worked through 2 of our 7. We've talked about physical barriers and phagocytes, and now we're ready to talk about immune surveillance or immunological surveillance. This is really orchestrated by those NK cells, which remember are natural killer cells. It is their job to move through the body and recognize and destroy cells that are abnormal. So this might be your cells that are abnormal, meaning cancerous or infected by viruses, but also bacterial cells will be identified as abnormal. So NK cells can also help just simply by removing pathogens. They're going to give us more of an immediate response when they identify this abnormal cell versus if we're talking about the T cell or the B cell. In order to activate T and B cells, there's a whole set of reactions that has to occur. It's pretty complicated, which is why we're saving it for Unit 3 discussion. So let's look and see at how NK cells are going to destroy identified abnormal cells. Oh, I hit the wrong button. Excuse me. There we go. All right, so remember our NK cells are moving through the body looking for an abnormal cell. And once they find it, they're going to get in proximity close to the abnormal cell and using exocytosis, release chemicals called perforins. And these perforins are going to perforate or poke holes in that abnormal cell. Once that cell membrane is compromised, you're going to have leakage of the inside out and the outside in. So this is a destroyed cell. So NK cells are attacking abnormal cells by the release of perforins. Next, so we're ready for our 4th type of non-specific defense or innate defense. Next is the interferons. Interferons are small proteins that are released by activated NK cells and T cells, fibroblasts, and also virus-infected cells. And we're going to see 3 different classifications of interferons discussed. Interferons are examples of cytokines. Cytokines are local hormones that are released. So this is an example of paracrine communication. When a cell is infected by a virus, it's going to release alpha interferons. And this is going to attract and stimulate NK cells. So those NK cells can demonstrate chemotaxis and move in the direction of the alpha interferon. And then remember, it's going to release perforins and poke holes in that abnormal cell. Fibroblasts are present at areas of injury because they're helping to rebuild that tissue. So here we see fibroblasts releasing beta interferons, and this will actually slow down inflammation in that area, controlling that inflammation because now we're ready for some repair. So this is a way of having a feedback mechanism on inflammation. And then our 3rd type of interferon is the gamma interferon. This is released by both NK and T cells, and it will stimulate the macrophage. So not only do you see NK cells showing up when you have a viral infected cell in order to attack those cells using perforins, but now you're also going to release the gamma interferon to bring those macrophages to the location to engulf and destroy possible pathogens. Okay, so we're ready for our 5th topic, which is the complement system. The complement system is a group of about 30 specialized proteins. They're complementing or enhancing antibody and phagocyte activity. So there is some additional information presented to you in your textbook. We're not going to get super detailed on this, but what we will see is that these proteins are going to be binding to your cells that have antibodies already attached or binding to foreign cells, and they can cause 3 things to happen. So that binding of those special proteins can trigger mast cells to release histamine, which we'll see is involved in inflammation. It can trigger cell lysis, so the rupturing of that abnormal or antibody tagged cell. or it can do what's called opsonization. And I had a student, several semesters ago now who said, oh, it's kind of like salt and pepper. It's making that pathogen tastier to those phagocytes. Opsonization is increasing the phagocytosis, making it easier for the macrophage to recognize and bind that it's a foreign cell that needs to be removed. Okay, and we're ready for our sixth topic, which is inflammation. Inflammation is a very localized tissue level response, and it's important because it's going to help to stop the spread of injury and also stop the spread of infection. And it's going to do this in a couple ways. So what we see happening is that if you have some cells that are damaged, they're going to be releasing some chemical signals into the surrounding fluid. So we're going to have what are called prostaglandins released, some proteins are released, and if that cell has ruptured, then potassium ion is going to be in a higher concentration than normal in the ECF. These chemicals signal mast cells to release additional chemicals. So the mast cells become activated and they're releasing more prostaglandins, they're releasing histamine and heparin, both of which are involved in inflammation. The prostaglandin is an important chemical that does stimulate pain receptors. So this is why often when we have injury in an area or inflammation in an area, it can trigger pain. The histamine and the heparin are causing inflammation by dilating your blood vessels. So that's increasing blood flow to that particular area. With the increased blood flow, you also get a little bit of a localized increase in temperature. So if you have like a local infection maybe in the skin, your area might look swollen, it might be red, and it might be a little bit hot to the touch. And also we're going to see an increase in permeability, which is going to trigger the swelling that's happening in these areas. Now, increasing your blood flow is important because in that blood, you're bringing clotting factors and platelets, so you're going to increase your blood clot. That increase in temperature right there localized can be important in increasing the enzymatic rates in that particular area, so we're going to see lots of benefits to this. Also, if you're increasing your blood flow, you're bringing along some additional white blood cells. So we've seen the importance of neutrophils, eosinophils, macrophages, and this ability to, kind of clean up. So to get rid of pathogens and cell debris and abnormal cells and all of that. So we'll see that also the release of the histamine and heparin, in addition to increasing your number of white blood cells simply by increasing blood flow, we have another example of positive chemotaxis. So these chemicals will attract neutrophils to the area. Neutrophils will release another chemical called cytokines that will cause more neutrophils to come to the area. So we have a positive feedback mechanism there. Those cytokines will also trigger an increase in the fibroblast activity. So that's going to be important in repair. And we saw earlier that the fibroblasts can release the beta interferon, which can then kind of shut down this process. So that's giving us a feedback mechanism, a way to return to normal. The last of our non-specific innate defenses is fever. Fever is defined as a temperature that is greater than 37.2 degrees Celsius or 99 degrees Fahrenheit. Now, in order to have an elevated body temperature, you have to have the chemical pyrogene released, which will reset your hypothalamus, which is responsible for your temperature control. So you're having to kind of shut down your negative feedback response that regulates body temperature. Now, those pyrogenes can be released from a couple of different places. Your cells can make pyrogenes, but also bacteria, mold, viruses, and yeast. So that's why we might have an elevated temperature when we have invasion of pathogens. Now, what's the benefit of the fever? We know having a really high fever over a long period of time can be dangerous, but allowing a low-grade fever to run its course might actually be beneficial. And the reasons are because, number one, the slight fever can inhibit some pathogens, so it might slow down their activity, while simultaneously increasing the body's metabolism so that our responses, our cell activity, our repair is all happening faster. This is an interesting fact. Changing your body temperature by 1 degree, so increasing your body temperature by 1 degree, is going to increase your cell's metabolism by 10%. So that is a pretty significant increase that might really go a long way in targeting the pathogens and repairing from that damage. All right, and that is the end of chapter 22, part 1.